BK channel. BK channels, big potassium, are large conductance calcium-activated potassium channels, also known as Maxi-K, SLUN or KK-on.1. BK channels are voltage-gated potassium channels that conduct large amounts of potassium ions, K+, across the cell membrane, hence their name, big potassium. These channels can be opened by either electrical means or by increasing Cat++ concentrations in the cell. BK channels help regulate physiological processes, such as circadian behavioral rhythms and neuronal excitability. BK channels are also involved in many processes in the body, as it is a ubiquitous channel. They have a tetrameric structure that is composed of a transmembrane domain, voltage sensing domain, potassium channel domain, and a cytoplasmic C-terminal domain, with many X-ray structures for reference. Their function is to repolarize the membrane potential by allowing for potassium to flow outward in response to a depolarization or increase in calcium levels. Structure. Structurally, BK channels are homologous to voltage and ligand-gated potassium channels, having a voltage sensor and pore as the membrane-spanning domain, and a cytosolic domain for the binding of intracellular calcium and magnesium. Each monomer of the channel-forming alpha subunit is the product of the KCNM gene, also known as SLUN. The SLUN subunit has three main structural domains, each with a distinct function. The voltage-sensing domain VSD senses membrane potential across the membrane. The cytosolic domain senses calcium concentration, Ca2+ ions, and the pore gate domain PGD, which opens and closes to regulate potassium permeation. The activation gate resides in the PGD, which is located at either the cytosolic side of S6 or the selectivity filter. Selectivity is the preference of a channel to conduct a specific ion. The voltage-sensing domain and pore-gated domain are collectively referred as the membrane-spanning domains. Within the S4 helix contains a series of positively charged residues which serve as the primary voltage sensor. BK channels are quite similar to voltage-gated K+ channels, however. In BK channels, only one positively charged residue, ARC-213, is involved in voltage sensing across the membrane. Also unique to BK channels is an additional serosegment. This segment is required for B subunit modulation and voltage sensitivity. The cytosolic domain is composed of two RCK regulator of potassium conductance domains, RC-cone and RCK2. These domains contain two high-affinity Ca2+ binding sites, one in the RC-cone domain and the other in a region termed the Ca2+ bowl that consists of a series of aspartic acid asp residues that are located in the RCK2 domain. The Mg2+ binding site is located between the VSD and the cytosolic domain, which is formed by asp residues within the serosome loop, asparagine residues in the cytosolic end of S2, and glutamine residues in RC-cone. In forming the Mg2+ binding site, two residues come from the RC-cone of one SLUN subunit, and the other two residues come from the VSD of the neighboring subunit. In order for these residues to coordinate the Mg2+ ion, the VSD and cytosolic domain from neighboring subunits must be in close proximity. Modulatory beta subunits encoded by KCNM-bone, KCNM-B2, KCNM-B3, or KCNM-B4 can associate with the tetrameric channel. There are four types of B subunits, bone-4, each of which have different expression patterns that modify the gating properties of the BK channel. The bone subunit is primarily responsible for smooth muscle cell expression. Both B2 and B3 subunits are neuronally expressed, while B4 is expressed within the brain. The VSD associates with the PGD via three major interactions. Physical connection between the VSD and PGD through the S4-S5 linker. Interactions between the S4-S5 linker and the cytosolic side of S6. Interactions between S4 and S5 of a neighboring subunit. Regulation. BK channels are associated and modulated by a wide variety of intra- and extracellular factors, such as auxiliary subunits BG, SLOBs, slow-binding protein, phosphorylation, membrane voltage, chemical ligands Ca2+, Mg2+, pKc. The pKa subunits assemble 1,1 with 4 different auxiliary types of B cell. Trafficking to and expression of BK channels in the plasma membrane has been found to be regulated by distinct splicing motifs located within the intracellular. In particular, a splice variant that excluded these motifs prevented cell surface expression of BK channels and suggests that such a mechanism impacts physiology and pathophysiology. BK channels in the vascular system are modulated by agents naturally produced in the body, such as angiotensin Roman-2 and Roman-2, high glucose or arachidonic acid A, which is modulated in diabetes by oxidative stress ROS. A weaker voltage sensitivity allows BK channels to function in a wide range of membrane potentials. This ensures that the channel can properly perform its physiological function. Inhibition of BK channel activity by phosphorylation of S695 by protein kinase CPKC is dependent on the phosphorylation of C1151 in C-terminus of channel alpha subunit. Only one of these phosphorylations in the tetrameric structure needs to occur for inhibition to be successful. Protein phosphatase 1 counteracts phosphorylation of S695. PKC decreases channel opening probability by shortening the channel open time and prolonging the closed state of the channel. PKC does not affect the single channel conductance, voltage dependence, or the calcium sensitivity of BK channels. Activation mechanism. BK channels are synergistically activated through the binding of calcium and magnesium ions, but can also be activated via voltage dependence. Ca2 plus dependent activation occurs when intracellular Ca2 plus binds. The binding site within the RC-cone domain has somewhat of a lower affinity for calcium than the Ca2 plus bole, but is responsible for a larger portion of the Ca2 plus sensitivity. Voltage and calcium activate BK channels using two parallel mechanisms, with the voltage sensors and the Ca2 plus bindings sites coupling to the activation gate independently, except for a weak interaction between the two mechanisms. The Ca2 plus bole accelerates activation kinetics at low Ca2 plus concentrations, while RC-cone site influences both activation and deactivation kinetics. One mechanism model was originally proposed by Monod Wyman and Chainjux, known as the MWC model. The MWC model for BK channels explains that a conformational change of the activation gate in channel opening is accompanied by a conformational change to the Ca2 plus binding site, which increases the affinity of Ca2 plus binding. Magnesium-dependent activation of BK channels activates via low-affinity metal binding site that is independent from Ca2 plus dependent activation. The Mg2 plus sensor activates BK channels by shifting the activation voltage to a more negative range. Mg2 plus activates the channel only when the voltage sensor domain stays in the activated state. The cytosolic tail domain CTD is a chemical sensor that has multiple binding sites for different ligands. The CTD activates the BK channel when bound with intracellular Mg2 plus to allow for interaction with the voltage sensored domain VSD. Magnesium is predominantly coordinated by 6 oxygen atoms from the side chains of oxygen-containing residues, main chain carbonyl groups in proteins or water molecules. C99 at the C-terminus of the serosone loop and 972 in the S2-S3 loop contain side chain oxygens in the voltage sensor domain that are essential for Mg2 plus binding. Much like the Ca2 plus dependent activation model, Mg2 plus dependent activation can also be described by an allosteric MCW gating model. While calcium activates the channel largely independent of the voltage sensor, magnesium activates the channel by an electrostatic interaction with the voltage sensor. This is also known as the nudging model, in which magnesium activates the channel by pushing the voltage sensor via electrostatic interactions and involves the interactions among side chains in different structural domains. Energy provided by voltage, Ca2 plus and Mg2 plus binding will propagate to the activation gate of BK channels to initiate ion conduction through the pore. Effects on the neuron, organ, body as a whole. Cellular level. BK channels help regulate both the firing of neurons and neurotransmitter release. This modulation of synaptic transmission and electrical discharge at the cellular level is due to BK channel expression in conjunction with other potassium-calcium channels. The opening of these channels causes a drive towards the potassium equilibrium potential and thus play a role in speeding up the repolarization of action potentials. This would effectively allow for more rapid stimulation. There is also a role played in shaping the general repolarization of cells, and thus after hyperpolarization AHP of action potentials. The role that BK channels have in the fast phase of AHP has been studied extensively in the hippocampus. It can also play a role in inhibiting the release of neurotransmitters. There are many BK channels in Purkinje cells in the cerebellum, thus highlighting their role in motor coordination and function. Furthermore, BK channels play a role in modulating the activity of dendrites, as well as astrocytes and microglia. They not only play a role in the CNS central nervous system, but also in smooth muscle contractions, the secretion of endocrine cells, and the proliferation of cells. Various G subunits during early brain development are involved in neuronal excitability, and in non-excitable cells they often are responsible as a driving force of calcium. Therefore, these subunits can be targets for therapeutic treatments as BK channel activators. There is further evidence that inhibiting BK channels would prevent the efflux of potassium and thus reduce the usage of ATP, in effect allowing for neuronal survival in low oxygen environments. BK channels can also function as a neuronal protectant in terms such as limiting calcium entry into the cells through methionine oxidation. Organ level and level. BK channels also play a role in hearing. This was found when the BKS subunit was knocked out in mice and progressive loss of cochlear hair cells, and thus hearing loss was observed. BK channels are not only involved in hearing, but also circadian rhythms. Slow-binding proteins SLOBs can modulate BK channels as a function of circadian rhythms in neurons. BK channels are expressed in the suprachiasmatic nucleus SCN, which is characterized to influence the pathophysiology of sleep. BK channel openers can also have a protective effect on the cardiovascular system. At a low concentration of calcium, BK channels have a greater impact on vascular tone. Furthermore, the signaling system of BK channels in the cardiovascular system have an influence on the functioning of coronary blood flow. One of the functions of the B subunit in the brain includes inhibition of the BK channels, allowing for the slowing of channel properties as well as the ability to aid in prevention of seizures in the temporal lobe. Bodily function level. Mutations of BK channels, resulting in a lower amount of expression in mRNA, is more common in people who are mentally challenged via hypofunction, schizophrenic, or autistic. Moreover, increased repolarization caused by BK channel mutations may lead to dependency of alcohol, initiation of dyskinesias, epilepsy, or paroxysmal movement disorders. Not only are BK channels important in many cellular processes in the adult, it also is crucial for proper nutrition supply to a developing fetus. Thus, estrogen can cause an increase in the density of BK channels in the uterus. However, increased expression of BK channels have been found in tumor cells, and this could influence future cancer therapy, discussed more in the pharmacology section. BK channels are ubiquitous throughout the body and thus have a large and vast impact on the body as a whole and at a more cellular level, as discussed. Pharmacology. Potential issues. Several issues arise when there is a deficit in BK channels. Consequences of the malfunctioning BK channel can affect the functioning of a person in many ways, some more life-threatening than others. BK channels can be activated by exogenous pollutants and endogenous gasotransmitters, carbon monoxide, nitric oxide, and hydrogen sulfide. Mutations in the proteins involved with BK channels or genes encoding BK channels are involved in many diseases. A malfunction of BK channels can proliferate in many disorders such as epilepsy, cancer, diabetes, asthma, and hypertension. Specifically, bone defect can increase blood pressure and hydrosaline retention in the kidney. Both loss of function and gain of function mutations have been found to be involved in disorders such as epilepsy and chronic pain. Furthermore, increases in BK channel activation through gain of function mutants and amplification has links to epilepsy and cancer. Moreover, BK channels play a role in tumors as well as cancers. In certain cancers, GBK, a variant ion channel called glioma BK channel, can be found. It is known that BK channels do in some way influence the division of cells during replication, which when unregulated can lead to cancers and tumors. Moreover, an aspect studied includes the migration of cancer cells and the role in which BK channels can facilitate this migration, though much is still unknown. Another reason why BK channel understanding is important involves its role in organ transplant surgery. This is due to the activation of BK channels influencing repolarization of the resting membrane potential. Thus, understanding is crucial for safety in effective transplantation. Current developments. BK channels can be used as pharmacological targets for the treatment of several medical disorders including stroke and overactive bladder. There have been attempts to develop synthetic molecules targeting BK channels, however their efforts have proven largely ineffective thus far. For instance, BM-SMINIS 204352, a molecule developed by Bristol Myers Squibb, failed to improve clinical outcome in stroke patients compared to placebo. However, there have been some success from the agonist to BKCA channels, BM-SMINIS 204352, in treating deficits observed in FMRO knockout mice, a model of Fragile X syndrome. BK channels also function as a blocker in ischemia and are a focus in investigating its use as a therapy for stroke. Future directions. There are many applications for therapeutic strategies involving BK channels. There has been research displaying that a blockage of BK channels results in an increase in neurotransmitter release, effectively indicating future therapeutic possibilities in cognition enhancement, improved memory, and relieving depression. A behavioral response to alcohol is also modulated by BK channels. Therefore, further understanding of this relationship can aid treatment in patients who are alcoholics. Oxidative stress on BK channels can lead to the negative impairments of lowering blood pressure through cardiovascular relaxation have on both aging and disease. Thus, the signaling system can be involved in treating hypertension and atherosclerosis through targeting of the subunit to prevent these detrimental effects. Furthermore, the known role that BK channels can play in cancer and tumors is limited. Thus, there is not a lot of current knowledge regarding specific aspects of BK channels that can influence tumors and cancers. Further study is crucial, as this could lead to immense development in treatments for those suffering from cancer and tumors. It is known that epilepsies are due to overexcitability of neurons, which BK channels have a large impact on controlling hyperexcitability. Therefore, understanding could influence the treatment of epilepsy. Overall, BK channels are a target for future pharmacological agents that can be used for benevolent treatments of disease.